Hello. In this video, we are going to discuss about a case study on lung chip mimics radiation injury. This case study was published in medgadget.com. So this is based on a research done by team of researchers. So now we will see about this research. Researchers at Wyss Institute at Harvard University have developed a microfluidic chip that can recreate some of the features of radiation induced lung injury. The lungs are very sensitive to radiation, and this can limit the application of radiotherapy to treat cancer. Accurately modeling radiation induced lung injury could assist in developing new methods to prevent and treat the phenomenon, but it has been difficult to study this before the advent of advanced organ on a chip models. The lung chip presented here contains human lung alveolar epithelial cells interfacing with lung capillary cells. The goal is to recreate the alveolar capillary interface, and then by exposing the chip to the radiation, the researchers can monitor the effects on these cellular populations in detail, as well as trying new treatments to reduce the effects of radiation. So the lungs are highly sensitive to radiation, with significant exposure resulting in radiation induced lung injury. This manifests as sustained inflammation and fibrosis, which can affect lung function. This can be issue for survivors of nuclear accidents who may have inhaled contaminated particles, but it can also affect patients undergoing radiotherapy, whereby dose limitation is required to avoid significant damage to the lungs. Okay. In any case, discovering how and why the lungs are so sensitive to radiation and trialing new treatments offers hope for such patients. The issue that it has been difficult to study phenomenon to date. Radiation induced lung injury is a complex condition and can vary significantly between patients based on variety of risk factors. Moreover, animal models of the condition do not typically recapitulate its complex presentation and entail serious ethical concerns. In response, these researchers have developed an advanced in vitro system that can mimic some of the aspects of radiation induced lung damage. So the chip that they have developed is a microfluidic culture system that contains human lung alveolar epithelial cells or in one channel where they are exposed to air as in like lungs, and another channel containing lung capillary endothelial cells that are exposed to a nutrient medium as a blood surrogate. This medium also contains immune cells as they are relevant in radiation induced injury. The two channels are separated by a semi-permeable membrane. The chip can be exposed to clinically relevant doses of radiation, and then cellular responses can be measured. So this is the diagrammatic representation of the chip that researchers have developed to model the response of lungs when they are exposed to radiation. And these are the DNA damage foci created by repair protein P53. Forming a better understanding on how radiation injury occurs and finding new strategies to prevent it poses a multifaceted challenge that in the face of nuclear threats and realities of current cancer therapies needs entirely new solutions, said by Donald Ingber, Wyss Institute director. The lung chip model that we developed to recapitulate development of radiation induced lung damage injury leverages our extensive microfluidic organ chip culture expertise, and in combination with new analytical and computational drug and biomarker discovery tools, it gives us powerful new inroads into this problem. So basically what the researchers in this case study they have done is they have developed a chip which mimics the lung function. They have developed this to study or to model the lung, right? What is the response when the lung is exposed with the radiation, maybe in terms of nuclear radiation or for the cancer treatment. So in the next video, we will see about another case study. Thank you.